Hi, everyone. Welcome to the fourth day of Garlic Week, part of the Winter Vegetable Sagra. Really excited to have you here today with Barbara Hellier, who is from the western part of hi, Barbara, of the National Plant Germplasm System here in the United States. She is in Pullman, Washington, and she's going to share with us a little bit about first the National Plant Germplasm System in general, and then a little bit specifically about where she works in Pullman. And we're really glad to have you here today, Barbara. Thanks. And you guys might recognize Barbara from Phil's presentation that on his excursion to collect wild garlic. She was in some of those photos. She was with him doing some of that work. So glad to have you here today.
Morning.
And but before we start, I want to say that Barbara and I were talking beforehand, and she mentioned a book, and someone else has asked about the book that Avram had talked about. So today, Barbara's mentioned this book called "Growing Great Garlic" from Ron Engelland, and it's from Chelsea Green Publishing. So that I think is still available online places. Also, it's great to buy books used. And Ted Meredith's book is the one that Avram mentioned yesterday, which is the complete book of garlic that he really likes. He did say that this one was going out of print, but someone else wrote in and said that you can get it as an ebook. So try to score those if you can, if you're really interested in learning more about garlic after being inspired by all of our garlic experts this week. Also want to mention that the garlic zine called Garlic Types and Market Niches that Avram wrote, that we published through OSU, and just it's online on eatwintervegetables.com. You can go there and find the whole thing. It's 12 pages. It talks about the different types of garlic. It has really great illustrations in it. And it's short and sweet. It's about 12 pages long. If you do want a printed copy of it, you can buy it on the Etsy site, the Culinary Breeding Network Etsy site, for $5, and we'll send you a copy just to cover the cost of that. It's really nice. It could make some nice stocking stuffers for garlic for garlic lovers, but we are really wanting to share garlic information and love this week through breeding, through growing garlic. When we talk about culture, we're going to talk later about medicinal uses, nutritional properties, folklore around it. So it's just a whole week to celebrate garlic, and we're really glad to have Barbara here today, who's going to know so much about garlic diversity that she's going to share with us. So if you have, I want to say that for the folks that are watching this right now on YouTube, I'm going to be watching the YouTube as well on a different screen over here. I will see the questions. Type your questions in. You might have to sign in to be able to do that. I need to check that. I'm sorry, I haven't. But people have been just writing in their questions, and I will field those to Barbara after we watch the 20-minute video that she put together for us. Sound good, Barbara?
Sounds good. Yeah. So the first video is about the NPGS system as a whole, and then I'll talk about our garlic collection.
Cool. Thanks. All right, we'll turn our screens off. Abba's going to play the video.
What we do here is we develop these collections of crop resources and their wild relatives. We maintain them.
And we normally store it in the form of seeds, but germplasm can take many forms. Our mission is to preserve this germplasm and then distribute it for research purposes.
This is important because researchers, breeders all over the U.S. and world, they request samples for research.
They can mine basically those genetic traits for anything that would be useful.
Some of these plants have more drought tolerance.
Or insect resistance or disease resistance.
We hope there's always something coming out from this material.
We have about 9,500 cultivated peanuts in the collection.
We have approximately 27,000 samples of grasses.
So we have about 30 rows back there. That's $30,000 worth of beans.
The pear collection is the largest germplasm collection here at the Corvallis Gene Bank. We are one gene bank out of many in the national plant germplasm system.
You know, our food security isn't really a given, and we shouldn't take it for granted. So we're preserving plant crop genetic diversity for today and tomorrow.
Welcome to the Plant Genetic Resources Conservation Unit in Griffin, Georgia. Our unit is part of the National Plant Germplasm System that is run by the U.S. Department of Agriculture. Our unit here is one of the 4 plant introduction stations that's part of the National Plant Germplasm System. The one here in the southeast in Griffin, Georgia. There's also one in Ames, Iowa, Geneva, New York, and Pullman, Washington. And those 4 sites will have a very diverse set of germplasm that's adapted to the regions where they're located.
Genetic diversity or the germplasm is the foundation for crop improvement. So a lot of this germplasm has diversity for all desirable traits that a peanut breeder would like to combine into a good variety.
What we aim to do is bring some of those seeds into the lab, put them onto the machine here, and this will give us a measure of the oil content of those seeds.
Today we're going to be working on sorghum. When I get an order, I'll start planting on the germ paper, and then I put it in the germinator, see if they're abnormal, dead, or diseased.
I'm packaging a warm season grass germplasm request. I take a sample of that plant, I wash the roots, and I wrap the roots in sphagnum moss and package it into bags.
We distribute about 50,000 samples a year to researchers all over the world, free of charge for research purposes. It's not really intended for home garden use. They're species that probably won't do very well in your home garden, but they're very important for plant breeders to use.
So here in Ames, we have about 54,000 accessions of more than 1,700 plant taxa. I've heard some people refer to the gene bank system as the thin green line between potential global starvation and food security. We're that backup.
We maintain spinach germplasm and a lot of other crops, and we have to extract the seeds so individually they have less dormancy. We want to make those desirable disease resistances and whatever else is there available to plant breeders.
The crops that the vegetable projects curate are pumpkins, melons, cucumbers, carrots, chicory, parsnips, and basil. Today we're working with pumpkins, extracting the seeds.
This is where we will select our 25 ears that are representative of this accession that are going to go through to imaging.
After we pick the berries, they're going to go through a common household blender. And then they'll be screened with water. After that, we can use an air column blower to separate the inert material from the seeds.
You can browse our entire catalog and see what's available and submit an order via a web cart. If that order is one that falls under our distribution policy of being for a research purpose or for breeding, then I print packets which are filled by our seed storage department to get that order to its destination.
Corvallis is a great area to grow plants. We are a gene bank that manages temperate fruit, nut, and specialty crops. We have scientists that work on the molecular analysis of the crops. They help us identify the particular cultivars and to see if we have unusual diseases in the plants. We all preserve genetic diversity of our assigned crops and work as a team to do so.
I like to compare myself to a librarian. Instead of being at a research library with books, research library is the plant materials, and we've got the trees lined up like books on a shelf, and I'm involved in several projects right now looking for better rootstock repairs, and that's where the wild germplasm is particularly useful.
Genetic diversity is important because we don't know what we're going to face in the future. So each of these plants, as more diseases are becoming spread around and more issues with climate change and soil conditions, having to grow on poorer soils. Some of these plants may have characteristics that allow breeders to introduce those traits into commercial quality cultivars so that we can grow in these changing environments.
This is called the Western Regional Plant Introduction Station, and we maintain a wide variety of material here in Pullman as sources of genes for adaptation to disease, to climate change, to environmental change.
So I'm just hand harvesting some peas in this greenhouse. We grow the peas inside to protect them from virus that they can get out in the field.
These are called isolation tents. They are primarily used to prevent cross-pollination so that we can keep the genetic viability and integrity of our collection.
Grasses are a major forage crop and also for turf. I maintain plots in the field, harvesting grasses, making sure the plots get weeded, check for insects, diseases, take data on them.
So you can see if you go around the greenhouse, there are a lot of beans that look very, very different, and it takes a lot of attention to make sure that they grow properly and produce lots of seed.
When I got here, I could not believe the amount of diversity that was maintained in a small location. It just blew me away. We have peas, lentils, lettuce, garlic, onion relatives, beans. The list is really large. I just can't emphasize how passionate most of the people that work here are and how much they love it.
Especially as the climate is changing around us, we want our plants to be adapted to these new conditions.
Here, there's so much diversity, you're always learning something.
I really, really like when we can identify that one unique accession that has really interesting traits in it.
The collection has just so much diversity, and a lot of it's visible. You can see the colors and things like that are in the collection.
I believe in the mission of maintaining genetic diversity for the future.
We have a responsibility not just to the U.S., but to the world.
I'm Barbara Hellier, and I'm a curator with the National Plant Germplasm System. The station I work with is the Western Regional Plant Introduction Station on the campus of Washington State University in Pullman, Washington. One of the crops that I work with is Allium. We work with all the species of Allium except for Allium cepa and Allium fistulosum, that's onion and bunching onion. So our collection contains garlic, leeks, chives, Chinese chives, and we have about we have accessions from 85 to 90, about 85 to 90 of the wild Allium species. 99% of the material that's maintained here in Pullman is stored as seed, and we store that in a storage vault set at 4 degrees centigrade and 30% relative humidity. The storage conditions for garlic are slightly different, so we've created, or we established a separate storage area, and that's down here, where down in the basement, where WSU and has growth chambers and also storage areas. So let's go down and take a look. This is our garlic storage facility. It's a temperature-controlled room that's set at 14 degrees centigrade and 40 to 50% relative humidity. Our collection is stored in these plastic boxes, and we've drilled holes in the side to minimize the amount of moisture that gets accumulated over the storage life. We currently have about 300 accessions in the collection. Our oldest material, the oldest accession that we have in the collection, is this one, and it was added to the collection to the NPGS in 1958, and that's about 4 years after the station was established. This particular accession is a hardneck variety that was originally collected or donated from the Farmer's Field family. The bulk of the collection was assembled by Dr. Richard Hannon. He was the former horticultural crops curator here in Pullman. He started assembling the collection in the early 1980s and continued into the late 1990s. The majority of the material that he added to the collection was from collections in Europe and from researchers in Europe. But we did add material from a small collection from Spain and Turkey. And he also added material cultivars from the U.S. and cultivars that he collected across the U.S. and in countries that he visited during his tenure. Our newest material was added from plant exploration trips, and primarily from Central Asia. So we have new material from Kazakhstan and Turkmenistan. The material from Kazakhstan was collected in markets, but the material from Turkmenistan was collected away from cultivation. We also have material that Dr. Simon and I collected in Uzbekistan. A few of those were collected in the market, but the majority of that material was collected away from cultivation. And we have a couple of accessions that I collected in Tajikistan. And again, those were ones that were collected away from cultivation. So the goal of the NVGS is to assemble collections that represent the genetic diversity of the crops that we are responsible for maintaining. We're currently working with Phil Simon to assess the genetic diversity of the garlic collection. In the past, how we assessed that was by assembling collections from across the species range, or wherever the crop was cultivated, and also looking at the morphologic and phenotypic characteristics of the material. So some of the characteristics that some of the data that we've assembled for the garlic collection are time to sprouting and storage, leaf morphology, allium and thiosulfonate content on a part of the collection. We also have scape or flower stalk formation and flowering characteristics for the hardneck varieties. What I'd like to do now is show you some of the garlic that we have in the collection. So the first succession I want to show you is one of the ones that was collected in Kazakhstan. So it's a nice purple striped garlic. It's got a really nice shape, I think. It's got more of a flat bottom. About 10 cloves per 8 to 10 cloves per bulb. It's really nice color. It does really well here in Pullman. The next succession is another one that was collected in Kazakhstan. It's slightly bigger and has a slightly lighter color. Again, it's a flat bottom bulb and about the same amount of cloves per bulb. This one is a really nice, consistent producer in Pullman. This is one of the accessions that was collected in Turkmenistan. It's a purple skin garlic, very few cloves per bulb, only about 4 to 5 cloves per bulb. On the larger size, the bulbs are larger. Okay, this is one of our longest storing accessions. It's a garlic, a soft neck garlic. In Pullman, it's on the smaller side. In our storage conditions, it'll store up to and over a year. This accession is one of our shortest storing accessions. It's originally from China. It's early maturing, and as you can see, it's already started to sprout. Because it's early maturing, we often
lose a lot of the bulb skins, but the skins are fairly thin. It would be a really good one if you wanted to have green garlic. So this next accession is one that was originally collected in Egypt, and it's a fairly small bulb with lots of cloves, usually around 20 cloves per bulb. It's got the lowest allium content and thiosulfonate content, so it's fairly mild. When it's, again, the size is when it's grown in Pullman. If it's grown in a warmer climate, they tend to the bulbs tend to get bigger. So this next accession is a garlic that was originally grown in Vietnam, originally came from Vietnam. It's a purple striped type, and in our collection it has the highest content of the material in our collection. It has the highest content of allium and thiosulfonate. So this accession is an example of a hardneck, and it's not anything that you can tell from the bulbs, but this particular accession is one that has really good flowering characteristics. I particularly like this particular garlic. I think it's got a nice shape, and it's fairly uniform. But the flowers it produces fertile flowers, and we've gotten true seed off of it. Okay, so this accession, although we don't have allium or thiosulfonate content data for it, according to our in-house taste tests, it's one of the more pungent accessions in our collection. And I particularly like this one because of the color. It's got a nice pearly color, and the shape, more teardrop shaped. This is another accession of that I particularly like. I like it's a soft-necked variety. It's got a nice uniform shape, and the skins are nice and white. It's originally collected in originally from Slovenia. Here's a composite of all the garlic that I just showed you. So you can compare the colors, sizes, and shapes. Well, thank you for watching, and I hope you enjoyed our short tour of the National Plant Germplasm System's garlic collection.
Cool. That was great, Barbara. Thank you.
My first attempt at video making, so
it was good. We the sound quality was a little hard there when you were inside the storage facility, but actually asked if we would reiterate some of that information, but we'll try to in these questions too. I have a question about when you were in there. I was thinking, I don't know if you mentioned this, but it was like, how many different accessions do you have? And I'm wondering how long, you talked a little bit about how some stored for certain periods of time, but like, how long do those store? And so that means like, how often do you have to grow each one out?
So all of our accessions are grown yearly. So they get planted in October, and we harvest, anymore it's early July that we're harvesting. Everything comes out of the ground. We do have some, like the one that I showed, the soft neck variety that I showed. We have a couple, maybe probably 4 or 5 that are soft neck. They're the tight skinned varieties that store for about a year, but most of the material doesn't store. It doesn't have they won't be viable in the following October. So everything, the whole collection gets planted every year.
Okay, that's been my experience. I just didn't know if you had a secret weapon over there.
No, unfortunately, we don't.
Okay, and how many different accessions are you growing every year? Is that like 300 or 400? Is it?
So we have about 300 garlic.
Mm-hmm.
So that's garlic and the wild garlic, where we call it Allium longicuspis. Some folks there's been molecular studies on that saying that the sativa and the longicuspis are the same, but taxonomically the taxonomists haven't actually officially said yes, they're the same. So we've kept them separately. That's about 300. And then we have, we'll have research material. And then we also have some material that we keep that are other species that are maintained as clones. So we have anywhere from 320 to 350 that we grow, allium that we grow every year.
Okay. And someone did ask if, are they grown outside or in a greenhouse?
So so the majority of the collection is grown outside. We do have a few accessions that Phil Simon gave us that he got from one of his collaborators from Bangladesh and Pakistan, Bangladesh mostly. And we haven't been able to grow those outside. We tried growing them in Pullman, and then we tried sending them down to Parlier, California, and they didn't survive. So we've tried to maintain them in the greenhouse. Garlic doesn't like being grown in a greenhouse. We haven't been able to figure out how to get garlic to thrive in a greenhouse. So unfortunately, we've lost a lot of that material.
Do you use, and people have asked this in past presentations, do you guys use like straw or do you use plastic? What do you use for, if anything, for weed suppression, or what kind of what kind of methods do you use for growing your garlic?
So we're not an organic, we don't do organic production. All of our, it's pretty conventional farming practices. We don't mulch. We haven't needed to. With the changing weather, we're getting some warm-ups in February where we're starting to get some sprouting. And so we might need to mulch in the future just to protect stuff, because we'll get the warm-up and then we'll get another cold snap. And that's in, we have a farm down on the Snake River. And when we planted the garlic collection down there, we've lost material. We've lost some of the cloves that we planted because they get a spring warm up and then they'll get another frost, heavy, hard, cold frost. So we might have to change our production methods. But right now we're not mulching. We fertilize in the fall. We have a split application, pretty typical. Apply some fertilizer in the fall, and then we'll do split application in the spring. So probably two applications in the spring. And from the previous, I don't know if the folks that were on when Avram was on, but our soil is so high in phosphorus, we generally don't add phosphorus. We'll add some nitrogen and a little bit of sulfur. Yeah.
Okay, cool. Thank you for doing
that. And we also, we don't irrigate too. All of our garlic nurseries are dry land, so we don't do any irrigation because we don't need to right now.
Beforehand too is there was a question I think during Phil's where we saw a lot of the specimens just growing out of rocks, and he also was talking about how there's not as much wild garlic in areas because it has become so dry. And you were saying that this is what garlic really does like and thrives in, is very well-drained soils.
Right. There's only a couple of allium species that I know of that will grow in a moisture, like a meadow type environment. All of them are, if they get water, it's in the spring, and then the soils get really dry. Yeah, all the species that I've worked with and collected in the Western US, they're all that way.
We do have several of the people that are on. I see their names. I don't know them, but I recognize them from earlier this week. And there have been a lot of questions about fertilizer, and someone's asking specifically what type of fertilizer that you use. And I remember she asked about nitrogen the other day too, which I didn't ask Avram, but what kind of fertilizer are you using?
Right. And I forgot to look that up. I don't know the specific formulation of nitrogen that we're using. It's yeah, I'd have to look that up. If she wanted to send me an email, I'd look that up for her. So
okay, Great. She's listening, I think. So send Barbara an email. You can find her easily online. So we have another question from someone who's been growing garlic. Here, I'm going to just read it out. He says, so I have some garlic that I keep in a food forest type bed. I used to harvest it every year and then replant in the fall. I stopped doing that about 6 years ago. The bulbs are now small and kind of remind me of onion sets, the little single bulb things. I'm wondering if anyone has any experience with this or have attempted to do this.
In a food forest?
Yeah, I'm not really sure. Yeah, food forest bed. So
you know, the Phil Simon showed the bulbs that we collected in Uzbekistan, and they were basically in a similar situation where nobody was harvesting them. The whole bulb stayed in the ground from year to year. And so that's my experience. If you leave your garlic in the ground, it's just going to get smaller and smaller and smaller. But when you harvest it and you break those cloves apart, so you give the individual cloves room to produce a good sized bulb, that's what's happening. You're just crowding out your planting material, basically.
So. Okay. Another question is, do you have any insight on soft necks? What happened there?
Phil is the expert on that one. My only guess is that somebody decided, they liked the taste of it, or they liked the size of the cloves, and there was a mutation that they found back, and yeah.
Yeah. And then he goes on to say, what are your thoughts on the most valuable traits in that group that would be useful for breeders if we could assess them?
Well, I guess my traits would be similar to what Avram said. They have tighter skins. They store, and I think that adds to the storage life.
Mm-hmm.
So, yeah.
Okay. Speaking of Avram, he sent in a couple of questions that I'm going to ask you. And he said, what morphological data is collected on the sessions within the collection and why?
So we've collected scape formation data. So I think he also mentioned that some of them, so we have the soft neck that don't produce a scape, and then we have some that are intermediate, so they'll produce a partial scape. I think Avram showed a picture of one that's an Asiatic that he had that had the bulbils in the stem. So they don't actually emerge from the leaves. The bulbils are covered by the leaves.
Mm-hmm.
And then we have the ones that produce, a full scape in the top sets and the umbel. So we've collected that kind of data on our collection, the leaf morphology, and it's mostly to characterize what the plants look like, so folks can use that data. We've also collected flowering information, so that we did a study for 2 years where we looked at the umbel formation, that's the flower head formation, and how the spathe opens, because that'll affect whether the umbel as a whole produces seed, and then whether the anthers were purple or yellow, and then how easy it was to remove the top sets from the umbel. So those are all characteristics. Oh, and then we also looked at pollen production. So production and then viability. So down the road, when all these folks that are interested in breeding garlic, if you want to do a controlled cross, then you can use a parent so like a Creole. One of the Creoles that we have, we never got true seed off of those, but they had a lot of fertile pollen. So they would be a male donor to do crosses with, one of these other ones that have, better flowers. So that was the impetus behind the flower characteristics. And then we also, the poster behind me is on top sets. So we have top set data and images for all of our accessions that produce top sets to look at size and color and number per umbel. And that's down the road, if somebody wants to use the top sets for a different production system, then they have that information. We haven't done any data. We haven't collected any data on the vernalization length.
Okay.
So we have sprouting data for some of the bulbs, but I haven't done it on the top sets. So
You did mention liking the flavor of particular ones. Do you guys taste them each also?
We don't do a lot of tasting. No, I did a really small taste test. And I know several people mentioned different methods to do garlic tasting. And what I used was I made hummus, because you use raw garlic to make hummus.
Mm-hmm.
And like what you mentioned, the flavor profile is very different. You know, you get some of them have a really strong garlic flavor up front,
Mm-hmm.
and then it peters out, and then others really it's mild, and then oh wow, then it hits you. You know, so yeah, I found hummus was another way to do a garlic tasting.
Yeah, I know that Avram does that. It's a lot of work to make that many hummuses.
Yeah.
But as far as like when I did it the one time, it was really hard for me to describe the different flavors, but the heat, like you're talking about, like when does the heat hit you? I always want to just draw like a graph that shows it builds, if it spikes and then crashes, like that's how I think of it. Really with all those garlics. So I don't know, maybe one day I'll do something like that. You talked a little bit about like if people wanted to do some breeding themselves and talked about some good candidates. And then you said also from Phil's talk, someone asked that, and you had a suggestion for one that you thought that a wild garlic that you would like to see people work with or that interested.
Yeah, so it's not a garlic, it's a different species of allium. And I thought somebody had asked about that, but I have some bulbs here. So it's Allium canadense, and it's wild. It's a North American wild it's a wild species of North America, and I just really am kind of in love with these bulbs. They have these nice pearly skins. I haven't tasted it yet, so I don't know what they taste like, but I just thought it would make a really pretty addition to a dish, even just whole, steamed or roasted. It's a species that has two varieties. One variety makes seed and one just makes top sets. It's not native to the Western U.S., so it's Midwest to East Coast, and all up in it's from Canada, Northern Canada, all the way down to Florida. So it's wide. It's not anything that's endangered or anything. So, but that's one that I thought would be an interesting candidate. As far as breeding, though, that would be start with the basics, and if you're really you're very ambitious and wanted to do controlled crossing, you could get some of these ones that have strong pollen production and have a lot of fertile pollen. And again, this data was collected, we collected the data in Pullman, and it's going to be different. Pollen production and viability is different. Temperature affects it a lot. So.
Yeah, that was another question that that Avram had. What differences do you see between the accessions in Pullman versus those maintained in Hellier, California?
Yeah, so we every year so that we have a backup of our garlic collection, we send material we send 10 cloves down to Hellier, California. The National Plant Germplasm System has a station down there, and they plant out the collection down there. And so the difference.
Sorry, where is that in California exactly?
It's close to Fresno, so it's in the Central Valley. It's south San Joaquin Valley.
Okay.
Yeah. And that's just because it's another repository that they can grow garlic there. They have irrigation, longer growing season, hotter temperatures. So we see quite a bit of difference in the size of the material that we get back from Hellier. I haven't done any profiles on like the thiosulfonate or alanine or the other flavor chemicals, differences between the two populations. A little bit difference on storage. The stuff from Parlier doesn't store as well. And Avram talked about northern vigor. So garlic that's grown in the north, you send it south and it tends to be more vigorous. I haven't ever had luck bringing material from Parlier and planting it in the field. If I needed to revive an accession from material from Parlier, I do it in the greenhouse, and then I get some plants, and then I'll plant those out in the field, do a spring planting. So it's, yeah.
Okay. Some more questions being written in are, what specific terms should we look for in the database to describe an accession that has good potential for true garlic seed?
Terms in the database. Actually, if you're interested in true seed production, send me an email. My email is on the website. You can look up the Western Regional Plant Introduction Station. Yeah, I have to make a confession here. We have the data. I need to publish it and then get it on the database. So it's stuff that isn't currently there. But if you're interested, send me an email and I'll work with you. That would be the best way.
Yeah. And then have you found garlic accessions where their colors are distinct from the white and purple spectrum already available?
Not really, though. We have the Creole. We have a couple Creoles that are red, very red, but no. Yeah, I've heard stories of true seed garlic producing blue bulbs, but I don't know if that's true or not.
No, it's not. Like I just recently, when working with someone from Hawaii, I've seen green and blue turmeric, which maybe you've seen, but we always see the yellow. And then we did this variety showcase event there, and it was like, oh my God, they had just come back from a collecting excursion in Southeast Asia and brought back. Amazing. Okay, do you have any insight on Allium leaf miner?
No, fortunately, we don't have it out in the West yet.
Oh yeah, I know. I was like, I don't know about this.
Yeah, it's an emerging pest. It occurs out on the East Coast. I'm not sure if the range is spreading or not, but yeah, it's been hard on the East Coast. Onion and I guess it's garlic.
Another question that Avram had too was what problems like pathogens arise that threaten the collection that you have there, like viruses or fungi, mites, and how do you manage that?
Okay, so we do have virus in our collection, and we're working to clean it up. We have about 8 accessions that we've sent through tissue culture, the meristem culture. We're working on another set of 10 now. It's a matter of personnel and time, funding to get this done. So we're working on cleaning the collection of virus, and as that happens, then the fungi will get cleaned out too. We know that we have some fungal infections. Fortunately, knock on wood, we haven't seen any white rot. We're really vigilant about keeping that out. It's we do have I've had some positive nematode tests for the soil. I've sent bulbs in for testing, and I haven't gotten any positive tests off of the bulbs for nematodes, which is fortunate. We don't see symptoms. And then dry bulb mite is a problem. It's becoming a problem all over. And that's one we got to figure out. I'm not sure. We treat all of our seed, the garlic cloves that we plant, for dry bulb mite, and we also treat with a fungicide pre-plant. The fungicide doesn't do much because the cloves are so thick it doesn't penetrate very far. But for dry bulb mite, we have found in just an in-house study that we did several years ago, that using the OSU information that you have on your website, we use a mixture of soap and mineral oil, and then they get soaked overnight. And that, when we did, we looked at material that we treated that way, and we found no live mites on that. And you really have to look hard. It's, fortunately we had a technician that could examine the cloves really well. Mm-hmm. We didn't, even down in the root crevices and stuff, we weren't able to find any live mites. So, but yeah, we need to figure out some fumigation for newly harvested material. So if anybody has any suggestions, I would love to hear it, because that's definitely,
yeah, it's Avram does talk about that a lot. It's a big problem.
Yeah, and I think he said that somebody's working on it in Cornell, so fortunately.
Another problem is funding. Sadly, there is not enough funding to preserve our genetic resources, like the work that you're doing. So what kind of funding keeps the collection going, and what would you like to see done with the collection if there were more resources available to you?
So we are federal, we're part of the USDA, the Department of Agriculture. We're in the Agriculture Research Service. Our funding comes from Congress, so we get appropriation funding every year. Fortunately, our funding has remained stable, but because it has remained stable or at the same level, our actual with inflation and with inflation, the actual dollars, the spending power of our money has declined. So yeah, it's that's our funding source. So we have appropriated money from Congress. So what
and what would you do if you had all the money that you wanted to?
Well, it would be great to, well, we're currently, I mentioned in the video, we're currently working with Phil Simon to genotype the collection, which is great. I mean, I know that Gail Volk did an AFLP study on part of the collection, and Phil's going to use SNPs, which is, so the SNPs, they take a snapshot of the genetic of the DNA in more places than the AFLPs. So, and he'll be able to, and we're also taking samples from all of the collection that we have now, which Gail's study only looked at part of it, because we didn't have all of it available for her. So yeah, I'd like to genotype the collection. I'd like to expand the allium and thiosulfonate. I'd like to do a more in-depth storage study to determine, because some of our material we could store at 1C and we get longer storage life, and some of it doesn't. It died. I tried it. It dies out at 1C. So I would do a more in-depth storage study. Yeah, I think those are the things off the top that I would like to see. There's always more work.
Yeah.
Dream, those experiments that you talk about, it's another study.
We could also do a massive tasting.
Yes.
Yeah. The hummus fest.
Yeah, I don't know how you do that. I mean, doesn't your palate kind of get saturated after a while?
No, I think so. I don't know if you heard how we did it. I mean, I didn't have any time to think about it really. Avram just gave me like 17 bags of garlic. So we came home and sat in my living room, and I had a chef come over and a couple of farmers and then just a couple of artists and myself. And we shaved it with a mandolin and put it in like the little, like the paper cups that you would put like ketchup in,
Yeah.
Grapeseed oil, since it's pretty flavorless relatively, and put it in there, and then tasted the full, the slice of clove, and then we could taste the oil too. I think mostly, actually, I think we first just stuck with the oil. It's like, let's just use like the infused oil. and we tasted that with bread. I mean, it's way too many to do. You never want to do more than like, 6 things at a time, it seems like. It's like, wow, whatever. We're not collecting data. We're just having a social time with garlic.
Yeah.
But it was very interesting. We wrote a lot of notes. I told Avram I would get to him, but I think that some people have asked for it on the chat on YouTube. I just haven't written it up, but maybe I'll just post a picture of our scrawls.
Yeah.
But I ordered garlic this year based on it. But what was so interesting when it was the top 3 that I really liked were actually from 3 different types of garlic. So it wasn't like I just liked one type of garlic. It was, for multiple. But
yeah, and I could see it if you're. I don't cook a lot, but I could see, depending on what kind of flavor you wanted, you would choose a separate, a different garlic. Yeah,
it's worth exploring more, I think.
Yeah.
Another question that someone had is, what determines if something gets accepted into the USDA system as an accession?
So we try not to have duplicates in the collection. Currently, I think we probably do have duplicates based on Gail Volk's study with the AFLPs. We probably have some duplicates, but we try not to have duplicates. We try to represent something that's unique. And it's hard with garlic, and it's hard because so many people share garlic, and they'll get they'll get a variety and they'll want to make it their own, so they give it a different name. So if you don't have good provenance information, it's kind of probably adding a duplicate.
How many excursion trips have you gone on to collect garlic?
So I've only done 2 for garlic. I've collected I work with a wide variety of material. So I also work with the beet collection, and I guess mostly my collecting trips have been beets. I've collected in Oregon for Allium, but two collecting trips for garlic, several for beets, and then legumes. I work with some of the minor legumes, so I've collected legumes. So, yeah, it's been some of the best. It's one of my favorite things to do is collect.
I bet it looks fantastic. I mean, I'm like, wow, this is a pretty dreamy job. I just went for the first time to look at radicchio farms in Italy in January with a group of like 22 farmers and chefs. And it was and we visited lots of seed folks and looked at how they grow there and like different ways that they use it, and ate a lot of radicchio in so many different ways. It's just so wonderful to go back to a place where there's a lot of diversity. I mean, this is, I feel like, what is the most exciting thing about, let's say, like the Variety Showcase, which is the event that has, been put on in person 9 times now, and hopefully we get to do that again maybe next year. I would love for you to be there. Taste garlic and breeding garlic, but it's really to show, like, to celebrate diversity in all these species that we eat. And be able to have people to have like the ability to talk to breeders and to talk to people that actually see, like yourself, see so much diversity that most people don't. Like, most people go to just a, mainstream grocery store. They're just going to see this one type of garlic probably their whole lives and not even know what the possibilities are and what exists, which is the same for every edible species, I feel like, that we eat,
Yes.
More.
Yeah. And it's, you do, because you go to the grocery store and it's not named, other than like apple cultivars. Now it's a lot better with apple cultivars. Yeah, it's great. But lettuce, I mean, there's a lot of different lettuce varieties. Even in Iceberg, we grew out our lettuce collection down in Salinas, and even among the Iceberg types, it's like there's a lot of diversity there. Genetically, they're pretty similar, but morphologically, there's some cool stuff.
Yeah, it gets people really excited. I really want to make one of those posters. I really like to make the posters to show the diversity, and there are really great ones that exist for garlic already.
Yeah.
Yeah,
I have one on my door. That's why I looked over at my door.
It's like to those and all of this type of work that you do is just really inspiring, I think, to people. So thank you so much for, preserving these very important resources for us in this country as well as the world. As you mentioned in your video, it's like this is something that's important for the entire world to preserve this.
Yeah, it's our it's been a privilege to be part of the mission here to do that over the years. So
that's great. So those are all the questions I feel like have come in. I've got text messages printed out from Avram. I've got the YouTube. I think that's everything. So do you have anything you wanted to add that wasn't mentioned today?
No, I think that's it. Thank you very much for inviting me. And yeah,
thank you so much. Thanks for all the work to put together the video and be here to present to us and tell us about what you do in your world.
All right. Well, thank you. Yeah, thank you for all your work too. It's great. I would be interested, if not as a member of the MPGS, just as a private citizen, to come. I would love to come to your variety showcase.
Great. Wonderful. Well, hopefully we do it next year.
Yeah.
Tomorrow is our last week of Garlic Week. We're going to have 2 speakers tomorrow. Actually, we'll have Erwin Goldman, who is a breeder at University of Wisconsin in Madison, Wisconsin. He is going to talk about nutritional benefits of garlic. And then after him, we will have Mary DiLorenzo Woods, who is an herbal folklorist, maybe folk or other way around, folk herbalist, who is going to talk about folklore and mysticism around garlic. So hope to see you guys then. This will be recorded as all of them were, and we'll see you tomorrow, hopefully.